Zende for giving me this opportunity to interact with the participants in this 4-week training program. So I take pleasure in meeting all of you and giving a short lecture on the given topic, that is intelligent packaging and its application in meat science. So I will share the slides now. Is it okay? Visible?
Yes, sir. Yes, sir.
My voice is clear?
Yes, clear.
Shall I proceed?
Yes, please go ahead.
Okay. So very good morning, dear participants. So the topic I'm going to present you is on intelligent packaging, the application in meat science. So if you see the trends in packaging in the latest time period, so mostly 4 important field is gaining importance. One is sustainable packaging, intelligent packaging, personalization on packaging, and transparent packaging. These are the 4 important fields of packaging which is gaining more importance, among which intelligent packaging is one of the most important growing trend, which is having a disruptive change in the field of packaging of meat and meat products. So why we should go for this intelligent packaging? That is foremost important. So if we see the first reason, we have to prevent foodborne illness that is going to be occurring due to consumption of spoiled meat. So already there are around 9,000 deaths in a year in the United States due to foodborne illnesses. So, and the next reason is going to be the minimization of food wastage. Like whenever we produce meat and if we see the availability, so the availability is there, but at the same time, there is some amount of meat or meat parts that is going at waste due to improper storage and preservation conditions, or we do not have a proper monitoring system in the stored meat and meat parts. So due to this, there is a huge wastage and we have to minimize this. So this can be done through intelligent packaging. Another thing is value for the purchase of the consumer. So whatever the money he is paying to purchase any food commodity, so he has to get the value for that. So what I mean is, suppose if he is paying a particular price for a fresh commodity, he should get the freshness in the product that is going to purchase. So he should not get a deteriorated food quality. So we have to ensure that. Another one is reducing the carbon footprint. The losses and waste, so which is also contributing to the increased carbon footprint. This can be minimized by using intelligent packaging and we can reduce the carbon footprint.
Participants, please don't interrupt in between. All of you keep your mic mute. All of you keep your mic mute. Otherwise, I will remove you from the meeting. One minute, sir.
Yeah, the next one is benefits of intelligent packaging. So why we should go for this intelligent packaging? One is the communication. So now if you see the trend, so there is a food package and you have a label on it and they have a lot of description on the food labels. And the one that is connecting the consumer regarding its quality is only the manufacturing and the expiry date or the best before date. So in most of the conditions, you can see that the actual date, expiry date, which is claimed by the consumer, the manufacturer, is not in reality, it is not really depicting the actual quality due to this if you are not maintaining a proper cold chain condition or there is electricity breakdown. So these are all the conditions which affect the stored meat and meat products. And we have to have another alternative so that particular type of indicator or sensor can communicate with the consumer about the stored meat and meat product quality than the expiry date or the best before date. Also, the consumers can use their smartphone to exactly know what is the quality or what is the traveling time or the storage period and all these things. And in order to trace back the product quality, so where it has come from. So it is also essential to have the intelligent packaging, so which will help in the traceability of the meat and meat products. So another one is connected packaging. So the consumer can either pay or know about the quality, or he can have some kind of a communication with the product through this connected package. So the communication is the most important thing. So which the product is going to give some kind of description about the quality and the related data to the consumers, so that he can purchase the product with much more guarantee and confidence. If you see the definition of intelligent packaging, so the intelligent packaging definition for any food commodity is a packaging system that is capable of carrying out intelligent functions. It may be a sensing, detecting, tracing, recording, and communicating. The objective is to facilitate decision making, to extend shelf life, improve quality, enhance safety, provide information, and warn about potential problems. So, if you see this definition, the extension of shelf life doesn't come under intelligent packaging. The extension of shelf life comes under active packaging. Other than that, all the other quality parameters, improve the safety, quality, providing information, warn about potential problems, these are all emphasized by the intelligent packaging. So for meat packaging through intelligence, we can devise the definition as to monitor the condition of packaged foods such as meat to capture and provide information on the quality of the packaged goods during transport and storage. This is the optimum definition for intelligent meat packaging. If you see the intelligent packaging, there are two types of modern packaging techniques. One is active packaging, another one is intelligent packaging. There is slight difference between both these packaging trends. If you see the intelligent packaging, if you have the food commodity within a package, so this food commodity has a headspace above this. So during its storage, the food is undergoing spoilage due to the microbial activity on it or some intrinsic factors which affect the food quality. So during this period, slowly there will be the utilization of the food components like carbohydrates, proteins, and fatty acids. Suppose if we take the example of protein, so the protein components will be changed into dipeptides and the much more components, smaller components like dimethyl amines, so and much more the amino acid products. So these products are going to be released on the headspace and they are available on the headspace. Now, if we design any indicator or sensor which can sense these components which are available in the headspace, this is going to be helpful for the consumer to indicate the quality of the food that is available within the package. So this food quality is communicated to the consumer. So this is intelligent packaging. On the other side, you have the active packaging technique. So in which there is some interactive material which is either incorporated into the food or which is available on the surface of the food or in the package, and which is going to enhance the quality, or this is going to interact with the food and increase the shelf. So this is called active packaging. So we have a clear-cut difference. The active packaging is going to preserve and enhance the quality of the food, whereas in the intelligent packaging, it is only communicating about the quality of the food product by having an indicator or a sensor in the head space. So why we have to go for this intelligent packaging, and what is its marketing potential? If you see the present trend, there is a huge market for this intelligent packaging. So it is about 48.72 billion US dollars forecast about in 2026. So this is growing at a trend of 4.15%. So really a huge market is existing for this intelligent packaging. And there are many factors which is contributing to this growing population and working population and increased demand for interactive and improved packaging solutions. So that's how this market is always growing, and the top market exists in the United States, followed by Canada, and some of the companies which are having a huge market include 3M, Avery Dennison, American Thermal Instruments, Temtem Corporation, SmartTrack, International Paper. So there are many companies which have a huge pull on this intelligent packaging. Also, the growing trend of nanotechnology also is adding a market growth for this intelligent packaging. So apart from this, there is a change in the printing practices, so which is also increasing the market for intelligent packaging through smart labels, photochromic inks, oxygen scanning sensors, and associated use of RFID technologies and printed electronics. If you see the global demand, so there is going to be 1.45 billion change in the next decade. So as I told you, the market is huge in US, followed by Japan, Australia, Germany, and UK. We see the topmost smart packaging startups. There are many companies. Mostly they are in Europe and many are in United States. And in Asia, we have the top manufacturers in Taiwan. So these are some of the intelligent packaging startups like Living Packets, MagicCAD, Algramo, EasyFlash, LineLoop, NanoLight. So what they do? So they mainly do with the intelligent packaging solutions. Some are like IoT enabled, Internet of Things enabled packaging devices. So some are using intelligent reusable packaging, and some are using these traceable models by the intelligent packaging. Some are using nanoparticles for developing the sensors, and some are using the connected packaging solutions in the supply chains. So if you see the intelligent packaging, so it has three broad categories. One is indicators, another one is sensors, and the other one is data carriers. Under indicators, we have freshness indicators, microbial indicators, gas indicators, or integrity indicators, time temperature indicators, so which also include the thermal abuse. So the other one is sensors, which include biosensors, electronic noses and tongues. So the last one is data carriers, which include RFIDs, radio frequency identification labels, barcodes, or quick response codes or QR codes. We'll see one by one. So intelligent packaging monitors the condition of the packaged food to give information about the quality during transport and distribution. So they may track, sense, or inform. So how does this intelligent packaging function? As I told you, meat is there within a package, and this meat will release some of the quality indicator compounds. It may be biogenic amines, some gases like hydrogen sulfide, total volatile basic nitrogen, ammonia, or any kind of gases. So which can be tracked by the indicators or sensors which are placed in the headspace on the inside of the overwrap. This will interact with the quality indicator compounds, and this will indicate some color change, and we can assess the quality, safety, and shelf life. So the other one is we can have time temperature indicators which are placed on the overwrap of the package, and this time temperature indicators sense about the external temperature, external environmental temperature of the food material, and this changes color depending upon the extent of thawing or change in the frozen condition or chilled condition, and they will develop some color change, and this will also indicate about the quality, safety, and shelf life. So any consumer, we can trace, track, or sense about the quality of the either chilled foods or frozen foods. Chilled foods can be through indicators or sensors, or frozen foods by the time temperature indicators. So as I told you, sensors, there are different sensors. So indicators, we have different indicators, and we have RFID tags. So we will first see the indicators. So the indicators are mainly used for indicating the freshness or the microbial quality. So they interact with the metabolites. The metabolite can be glucose if it is from the carbohydrate metabolism, or carbon dioxide, ammonia, dimethylamine, or trimethylamines. They may be protein breakdown products or the biogenic amines. So we can target any metabolites to indicate the freshness. So they may be qualitative, or they may be visual, or they may be semi-quantitative. So they will change color, and they will give indication about the quality. So the time temperature indicators are also used. So they are also visual sensors. So there are several studies on this. So the time temperature indicators functions on mechanical type, chemical type, or enzymatic type. Oxygen indicators, they mainly functions on the redox types. So it may be a reducing agent or oxidizing agents. So we also have some of the indicators for carbon dioxide, microbial indicators. So they may be a dye-based or pH-based. We have pathogen indicators. Some are chemical-based, some are immunochemical-based, and some also interact with the toxins. So first we'll see the freshness indicators. So the freshness indicators are based upon the metabolites. As I told, it may be a glucose, volatile basic nitrogen, or biogenic amines, or ATP degradation products. It may be like xanthine, hypoxanthine. We can target anyone. So several research have been conducted. So like one is like pH sensitive material. So it was used for chicken, pork, and beef spoilage sensors. A freshness indicator is developed based upon the chitosan, cornstarch, and hibiscus rosacea and anthocyanin. This again for chicken meat. So likewise, many freshness indicators have been developed. So these are all mainly the indicators quoted onto the labels, and these labels are stick onto the inside of the overwrap, and when the particular amount of metabolite is present, they react with these indicators, and they produce color change. Like we also have total volatile basic nitrogen indicators, which are based on starch, polyvinyl acetate, and rosal anthocyanins. And likewise, several total volatile basic nitrogen indicators are available, which are targeted for the pork, chicken meat, and poultry meat. So normally the total volatile basic nitrogen concentration in a fresh meat is less than 20 milligram. If it is increasing more than 20 milligram, so then the color is changed in the indicator and the quality deterioration can be assessed or the spoilage can be monitored. So other ones is gas indicator. We can also have a sensor for carbon dioxide or hydrogen sulfide. So like we all know the myoglobin, if it getting deteriorated, or the myoglobin if it is going to be broken down into its byproducts, the hydrogen sulfide concentration is increasing in the food material or especially the meat, and this increase in the hydrogen sulfide concentration can be trapped by the indicators and the freshness can be assessed. So indicator of gas and integrity. So we can also indicate about the gaseous concentration, whether carbon dioxide is more or whether the sealing is proper or not. This is about integrity. If the sealing is not proper, suppose the particular oxygen concentration in a package is going to increase, and this can be assessed by having some oxygen indicators or gas indicator for oxygen. So likewise, we can have some redox dyes. Like initially it will be colorless and it will change to blue color when the oxygen concentration is increasing in a package atmosphere. So this is mainly methylene blue, so which changes its color upon interaction with oxygen. Same thing, initially it is light pink color and it changes to blue color upon oxidation. So we can also have microbial indicators. These are all diamond based sensor system. So like if there is more presence of lactobacillus, so because of the presence of diacetyl vapor, the color gets changed and we can assess the microbial quality. The other one is time temperature indicators. As I told, the time temperature indicators are nothing but they change the color depending upon the exposure duration other than the actual permitted period and temperature for a particular commodity. Say suppose meat is frozen at -20°C. If you are not maintaining the storage temperature for a particular duration. So if the thawing duration is more than 4 to 5 hours from its actual temperature, that is -20°C. So this is going to slowly change the meat quality, and the ice crystals present in the meat is going to melt, and this is going to increase the fluid around the meat, and also the microbial load in the meat is going to increase as the time duration is increased, and this is going to drastically affect the meat quality and the even the nutritional quality of the meat. So we have to have some time temperature indicators which will indicate how much duration of thawing has occurred and how much quality deterioration has changed in this thawed meat. So for this purpose, we need to have time temperature indicators. So there are several time temperature indicators available, so which are mostly based upon enzymatic indicators or chemical indicators. So what happens is these enzymes which are present in these indicators, they remain dormant and inactive when the frozen condition is maintained. So when the thawing is going to occur, the inactive enzymes or the dormant enzymes, they will get activated and they will try to utilize the substrate present in the indicators, and thus there will be either depolymerization, depolymerization or any other change, the oxidation, reduction, any change will occur, and this is going to change the color of the enzyme and the substrate color. So this is going to produce a change in the indicator, and thus you will have a color change due to thawing. So the consumers can assess the product was thawed or not, and it is essential to have irreversible color change, because the manufacturers or the retailers can cheat the consumer, because if we have a reversible color change, then the initial color, it will go back to its initial color, and you cannot assess whether it is thawed or not. But once you have a irreversible change, suppose from yellow color, or say suppose here light pink color, it is going towards the pink color. So this is one irreversible color change. So we need to have this irreversible color change. So that if thawing has occurred, even if you are refreezing it, this color will be retained, and the retailer or the manufacturer cannot cheat the consumers. So we are also having some of the time temperature indicators based upon the thermochromic inks. So these are all the commercial inks based upon the chemicals, so which are printable. So these thermochromic inks can change its color upon a change in the temperature or storage temperature, especially the freezing temperatures or the chilling temperatures. So the other one is, so far we have seen the indicators. So the indicators may be freshness indicators, gas indicators, time temperature indicators, or the pathogen indicators. So the next one is sensors. So the sensors are a combination of the transducers and the detectors. So here certain analytes are released in the stored meat, and these analytes have to be captured by the transducers, and the signals will be transmitted, and colorimetrically or chemically or electronically we can assess whether the food is spoiled or deteriorated or not. So we need to have electronic system within these sensors. So the first one is biosensors. So we have some research into this, like they have used colloidal gold or palladium nanoparticles, and they made a strip. So this strip is sensitive to Listeria monocytogenes and Klebsiella, and when this is also coated with the antibodies specific to Listeria and Klebsiella. So when this antigen is reacting with this antibody, it produces color change, and thus we can have these biosensors specific to Listeria monocytogenes within the package. So this is nothing but lateral flow acid strips which are stick onto the inner surface of the package, and when this food material is in contact with this lateral flow strips, it produces color change, and qualitatively we can detect whether Listeria monocytogenes or Klebsiella is present in the food material or not. So we also have some kind of sensors, so which can detect the pathogens present inside the food package. So these are some of the labels, so which change color, like so if it is fresh, it is tangerine in color. If it is not fresh, the color fades up. We also have some of the indicators or sensors for toxins, like against salmonella toxins, or E. coli or Listeria or Campylobacter. So the toxins, we have certain antibodies which are in contact with this packaging material, and the bacterial toxins which which will come in contact with these antibodies, so they will produce the visible color change. We also have food sentinel system. So the food sentinel system is nothing but this is a barcode system. So in one of the barcode, they change with this the pathogen detection system. So one of the code will be having a pathogen detection system in which the antibodies specific to the pathogen will be coded. So it will be having the substrate, antibodies, and the other things. So which again will interact with the pathogen and it will produce color change. Even we have sensor for antibiotic residues. Like again, it is based upon the immunochromatographic system or lateral flow system against ciprofloxacin residues in beef meat. So this is sensitive even to 100%. So the minimum detectable level is 25 milligram per kg. This produces color change within 1 minute. So this is again lateral flow system, so which is specific to ciprofloxacin. So the second one we have seen is sensors. The first one we have seen is indicators. The second one we have seen is sensors. So the third one is data carriers. So these are all coming under intelligent packaging. So the data carriers are nothing but these are the devices which carry a huge bulk of information related to the food or the meat or meat product. So this may be a barcode, this may be a QR code, or this may be one RFID, radio frequency identification system. So they have a huge amount of data within them, and this can also be used as intelligent packaging system. So among these, the first one is connected packaging. What is this connected packaging? Like we can have quick response code, so which is printed on the label, and we can use a near field communication, like we can scan the QR code, and by which the consumer can get lot of information about when the product was manufactured, and what is the storage temperature, whether how it was processed, and what are the information for use after you purchase, and what are the nutritional information, and what is the quality, what is the quality, whether it is fresh or not, what is the microbial status. So we can have lot of information within this QR code or near field communication label present on the package. So this gives a lot of authentication and confidence and guarantee for the consumer to purchase the meat and meat products. So there is also a reference paper on food barcode for the chicken meat. So the other one is RFID sensors. So the radio frequency identification sensors have also been developed for the meat. So this is based upon printed conducting polymer PDOT, that is PDOT PSS, polyethylene dioxythiophene, polystyrene sulfonate, and this is a mixture of two ionomers. One is PDOT and PSS, and this is integrated with IoTs, Internet of Things. So what happens is, so there within the package, so suppose meat is present, and we attach the RFID tags. So this RFID tags will have all the informations about the storage temperature, the meat, about the meat, its quality, its nutritional status, its manufacturing status, expiry, and how to use after you purchase, how to dispose the package, after you purchase, all the information should be printed on this RFID chip. And during the transport of the food material, we can monitor this change in temperature or change in quality, and where is the meat or where is the package at a particular time or duration during the transit by sitting at a particular place. So at a point, we can know about the complete quality of the particular food commodity by sitting at a place, and we can monitor the quality of the food commodity through these RFID sensors. So we have RFID labels for meat freshness and traceability, and we also have RFID label for oxygen indicators. So this is used in map system for pork freshness. So this helps in a lot of functions, like we can code a lot of things into this, and a lot of companies like Epsila, RFID enabled solutions, HRAFN Limited Sweden, they all are producing RFID tags for meat and fish. So this is about different intelligent systems. So one is indicators,
Please don't
sensors,
talk in between.
other one is data carriers. So we also done some of the research on these intelligent packaging systems, so which I want to just share with you. So this is on buffalo meat. So we have developed freshness indicator strips for buffalo meat. So this was developed by Dr. Vivek and our team when he was doing his research in IVRI. So like the buffalo meat, when its quality is fresh, so it has the sensor strip, it is yellow in color, and when its quality deteriorates, the color changes from yellow to blue color. So this we have also compared with many meat quality parameters and the microbial quality. So when the color changes to dark blue color, the microbial count exceeds 7 lakh CFE per gram. So we can say that it is a good indication about the microbial quality as well. So the next one is time temperature indicator for buffalo meat. So this is based upon enzymatic reaction. So we have integrated amylase and starch in the sachet type. So this is attached to the packages, and when this meat is kept at proper storage condition, that is frozen condition, so it is the color is not changing. So once you expose for a very long duration above the actual freezing condition, then the color deteriorates and the meat quality also deteriorates. So the color changes from dark bluish black color to yellowish color. So this is another time temperature indicator which has been developed for chicken meat. Initially, the time temperature indicator, which is made up of lactase and guaiacol. So this is colorless when the freezing condition is maintained. So if you are not maintaining the freezing condition properly, then the thawing occurs in the meat, and the enzyme gets activated and it tries to utilize the substrate, and the color change occurs, and the color changes from colorless to a pale brown color and dark brown color if thawing is more, and you can appreciate here a huge drip loss, and the meat quality also gets deteriorated, and the color of the indicator changes to dark brown. So this is another time temperature indicator, so which has been developed for vacuum packaged chicken sausages. So if you are storing the vacuum packaged chicken sausages at proper freezing temperature. The time temperature indicator, this is made up of urease and carbamide. So this is yellowish in color if you are maintaining proper storage condition. If you are not maintaining proper storage condition, it changes to pink color, and you can see that you can clearly appreciate the thawed sausage and the frozen sausage. This is another intelligent packaging system which we have developed for luncheon chicken sticks. Here we have developed a film, so which is incorporated with anthocyanin and carboxymethyl cellulose, and this film is a chromogenic or color changing film. So initially it is pink in color, and you can also appreciate the product color during the initial stage. So from the dark color, the meat product color also changes to a light color, and even the color changing film. The film from the initial pink color, the color fades away as the quality of the product deteriorates. It is used as a wrapping material over the product. So this packaging film, the intelligent packaging film, gets oxidized when the product quality deteriorates, and the anthocyanin present in this packaging film gets oxidized because of the phenolic group that is present in this film. So the next work which we attempted is silver nanocomposite based intelligent packaging strip. So this is based upon silver nanoparticles and titanium oxide, so and anthracene. So this is initially pink in color in a chilled chicken meat, and if you are storing for a longer duration, the color fades away and it changes to brown color. So this is again indication of the increase in the metabolites, especially it is sensitive to ammonia. So the other work that we conducted is gold nanocomposite based intelligent packaging strip. So here this is based upon anthocyanin, curcumin and gold nanocomposite based strip. This again sensitive to volatile metabolites, especially total volatile basic nitrogen, so which is being released in this stored meat, and this interacts with this strip and it changes to bluish color when the meat quality deteriorates. The other work that we have done is to develop an enzymatic nanocomposite based time temperature indicator for frozen chicken meat. So initially this time temperature indicator strip is colorless. So in a frozen meat, this is not frozen. So this is a frozen meat. You can appreciate here. So when it is thawed to 5 degree and 10 degree, if you see, if you are thawing to 10 degree for a longer duration, so there is a drastic difference in the change in the color of the sensor strip from colorless to brown color. And if you are refreezing, again the color is irreversible. The retailer or the wholesaler cannot cheat the consumer that the meat is already undergone thawing, and you can appreciate if any thawed meat, excessively thawed meat is again refrozen, you can visually appreciate the color change and you can say it is a thawed and refrozen meat. So this is about the various research conducted on intelligent packaging. We see the challenges and opportunities. We have a lot of opportunities, like we can, there is a lot of waste reduction because of the intelligent packaging, because we can continuously monitor the quality of the meat or meat products that is stored in the chiller or the freezer, and we can ensure that the meat or meat product doesn't go waste. So we can effectively monitor the cost, and it is profitable for the manufacturer or the retailer, and it gives lot of information for the consumer. So he can confidently purchase the meat and meat product. The another field is thin film electronics. So this is an upcoming field. So this is again giving us another added advantage to work on this field. Another one is nanotechnology. So this is also another upcoming field. So this is also giving us ample scope for working on this intelligent packaging. And also real-time capabilities. We can monitor the quality on real-time basis. So this is giving another added advantage. The cyber security is also another the opportunity for the manufacturers and the retailers or the wholesalers, even for the consumers. And also it gives new business model for the manufacturers. But the challenge here is, so how to fabricate these indicators or the sensors, and how to bring about these indicators or sensors or the data carriers without adding much cost onto the packaging expenses. So already in the industry, they invest about 5% at least on the packaging cost. So we have to see, if you are investing on this intelligent packaging, again the cost is going to increase on the packaging. So how far we have successfully incorporated this intelligent packaging without incurring much on the packaging cost. So at least we can aim at, say, increasing the 7% on the packaging. If you are going for 10% and more, so it is going to be a burden on the manufacturer. So this is another challenge for the persons working on this intelligent packaging, how effectively we are going to fabricate intelligent packaging devices. The other challenge is disposing of this intelligent packaging systems. Already the packaging of the plastics is posing environmental challenge. So how we are going to produce the packaging devices which are environmental friendly for this intelligent packaging. So these two are great challenging for the developers or the manufacturers or the researchers involved in this intelligent packaging. So the future of intelligent packaging is evolving, and it has pretty interesting future. And in future, we are going to see that this intelligent packaging is going to directly talk with the consumers. So because of the increase, the technological advances like augmented reality and the artificial intelligence, once you take the package in your hand, and just if you are going to scan with your smartphone, the package is going to talk with the consumer because of the embedded technology on it. So that is the such advances that is going to happen in this intelligent packaging field. So this is going to be more customized and personalized too in the future. So as I said, so because of this artificial intelligence and augmented reality, any person, they can even by sitting at a place, he can just see what is happening in a particular company, or what is happening in a particular wholesale unit or particular supermarket, how the food is stored and what is the storage temperature. He can interact with the people in that supermarket. He can interact with the marketing people. He can interact with the CEOs or company president. So such is going to be the development in the intelligent packaging. So this is going to be really interesting in the future. So as I told you, the safety concerns and limitations are also there with this intelligent packaging. So we have to see whether the packaging material are conforming to the standards laid down in the various countries. So we have to adhere to the legislations and restrictions. So whatever we use in the intelligent packaging, whether they are of food grade or not, whether they are sustainable for the environment or not, whether they are ecologically friendly or not. So we have to see all these things, and we have to consider this safety and regulations existing within the countries. And we also should see whether the sachets or the strips or anything, whatever you use as an intelligent packaging material, whether we are avoiding the accidental leakage or we are preventing the migration or not, whether we are taking proper care to prevent the migration of the material, the intelligent packaging material into the food material or not. So we have to see all these things. So these are all the limitations and also the safety concerns posed on intelligent packaging. So already there are regulations in different countries, like in US we have Environmental Protection Agency. So they have waste reduction model, so which is having a document on how to use this packaging materials for intelligent packaging as well as the normal packaging. And we should also see how we can reuse and recycle. This is another limitation. So we have seen intelligent packaging takes the form of indicators, sensors, and data carriers. So intelligent packaging as an indicator may be freshness indicators or spoilage indicators, or it may be gas indicators or time temperature indicators. Or we can use this intelligent packaging in the form of sensors. The sensors can be radio frequency identification devices, RFID sensors, or it may be in the form of QR codes. So we can also use these indicators in the form of data carriers or some sensors for the microbial quality or the toxins, or we can have some sensors for the even the residues for the antimicrobials like ciprofloxacin, or some of the microbes like we can use for E. coli or Listeria or Klebsiella. So this intelligent packaging has vast application. So I would like to conclude this talk by telling that this intelligent packaging provides customer with a connected, informative, and entertaining experience while ensuring brand transparency to the consumers. So intelligent packaging is completely functional and reliable system, and it provides an enormous ground for both fundamental and applied research for the years to come. Intelligent packaging and connected packaging has a remarkable feature, and it provides vast scope for the researchers to work upon, to bring this intelligent packaging into reality in all the packages that is commercially available for the consumers, so that all the consumers can confidently purchase the product and they can be pretty sure about the quality and safety of the food products they consume. Thank you one and all, and I thank the organizers for giving me this opportunity. Thank you.
Thank you, Dr. Kandipan, for excellent presentation and very recent and updated information you have provided on intelligent packaging in different meat products. Very recent informations and what are the challenges and opportunities in particularly on in the aspects of the intelligent packaging. Now, the one or two questions from the participants, please.
Participants, you can post your questions in the chat box or you can ask.
Anybody want to ask any question? One question is there.
Yes, there is one question. Is there any controlling monitoring agency in India which specifically looks at the packaging quality of food products? Yes, in India we have Food Safety and Standards Authority of India packaging regulations. We have special packaging regulations, so which gives a detailed information what are all the packaging material we have to use and what are the standards for each packaging material, and even what should be the migratory limits, that is also given. So you can see FSSA website, and you can download the packaging regulations, even the labelings. Packaging, it is both for packaging and labeling. So labeling regulation is also has a detailed information how to label it, what are the informations you have to incorporate, and even the limits like what is the trans fat limit and what is the limit for say the other things which has harmful effect on the health of the consumers. So everything is there. So you can download the material and you can have a look on it. So FSSA has regulatory limit for that. So the other one is antimicrobial packaging. Does it has any antimicrobial in laminates? Yes. So you can incorporate any antimicrobial agent directly into the packaging films. So once it is getting manufactured, like you can use either any natural antimicrobial, like you can use essential oils, like you can use cinnamon oil or thyme oil. So these are all natural. Or you can use any synthetic antimicrobials, any chemical based which is permitted. So previously we are using any even some acids. Okay, so like even any acids which are food grades. So which, those can be used, like sometimes citric acid, although it is more of regulating acidity. So some has some acids which has you can use lysozymes which has antimicrobial activity, or some uh, mm, which are from some lactobacillus. They are also produce some antimicrobial peptides. They can also be used as antimicrobial agents. So they can be either directly infused into the packaging material and you can get the laminates. This is one aspect. The other one is you can have the packaging material, you can either spray or you can either put some antimicrobials on its surface and you can pack the food into that. So this is another aspect. So by this you can use antimicrobial as packaging devices. So the other one is future of biodegradable packaging in meat industry. So there are already some biodegradable packaging materials, but they are commonly used for other food materials. We are also developing a biodegradable packaging material, especially for meat. So because meat contains more than 70% moisture in it, so the available biodegradable packaging materials are soaked when in contact with the meat. So we need to develop a biodegradable packaging material, so which resists this soaking when in contact with the meat. So other one is developing the sealability of the film. So this is another challenging aspect. So whether the film is sealable or not, although it is biodegradable. So the other point is so resisting the water permeability. So it should be water impermeable. So we have developed a film in our lab, so which is biodegradable as well as which are equivalent to the quality of the low density polyethylene film. It has all the barrier properties and also the biodegradability aspect. So it has a good feature in terms of Indian food market because if you see the food safety regulations for this packaging material, now we have to, in the first phase, we have to contain the usage of single-use plastics in the next 5 years. So next, we have to completely eradicate the usage of plastics. Then the biodegradable packaging material are of great use and it has good feature. So next is any packaging company has commercial capability in India to use this antimicrobial. So far in India, they are not producing antimicrobial packaging film, but we can produce. Even in our lab, we have produced this biodegradable packaging material, which also has antimicrobial activity in it, but we can produce.
Thank you, sir. Thank you, sir, for clearing all the doubts of the participants. So thank you very much, sir, for sharing your valuable information and providing the excellent lecture. I think sir is the first in India to start this intelligent packaging application, especially in the meat industry. So thank you, sir, for your presentation. And thank you all the participants for asking questions. Now, will we start with the next speaker?
Yes, thank you, dear participants, for interaction. And I thank from bottom of my heart Dr. Vivek and Dr. Vilas Vaidya for giving me this opportunity for delivering this lecture and interacting with the participants. Thank you so much.
Thank you, sir.
doctor.
Thank you, sir.
Thank you.
So now we have the next speaker, next eminent speaker for the today's session, Dr. Prem Singh Yadav, sir. Sir is.
पहले देख लो सर है क्या?
Sir, are you there, sir? Yadav, sir. I think sir is here. He has joined, but I think he came. Sir, good afternoon, sir.
Hello, sir. Sir Yadav, sir, good afternoon.
You are listening me?
Yes, sir.
Yes, sir. Yes, sir.
Yes.
Okay, good.
So, good afternoon, sir. So now we have the next speaker, next eminent speaker for the today's session, Dr. Prem Singh Yadav, sir. So, sir is working as a principal scientist and head, APR division, ICR CIRB, Hisar, Haryana. And sir will be delivering his lecture on animal cloning application and status in India. So, before we proceed for the lecture, I will request Dr. Vaidya, sir, to give a brief introduction about Dr. Yadav, sir. Yeah, Vaidya, sir, please.
Thank you, Dr. Vivek. Dr. Prem Singh Yadav, sir, he is working as principal scientist in animal physiology and reproduction division, Central Institute for Research on Buffaloes, Hisar, Haryana. Dr. Prem Singh Yadav, born on 10th April 1963 at village Nimot, Tehsil and district Rewari, Haryana state, India. He completed MSc and PhD from Hisar Agricultural University and postdoc from Germany in the year 2003 and 2004. Presently working as principal scientist in APR division, Central Institute for Research on Buffaloes, Hisar. Dr. P.S. Yadav, he is worked as team leader and have been working on assisted reproductive technologies in buffalo, including cloning. In the year 2015, CIRB became India's second institute by producing a cloned buffalo bull named Hisar Gaurav. In 2017, the team had cloned an Assamese buffalo bull in the field away from the institute. Under his leadership, ICR-CIRB Hisar produced a total of 10 cloned buffalo cows during 2015-2020, and this achievement of producing maximum clones is recorded in India Book of Records. Produced 7 clones from a single superior bull M29 are and recloned an earlier cloned bull Hisar Gaurav buffalo. In addition to buffalo cloning, the team has also been working on semen biology, in vitro embryo production, that is IVF, transgenic gene edited embryo production, embryonic and adult stem cell research, and embryo transfer in buffalo. So, sir has many achievements, and it's an honor and privilege to all of us that sir is with us for the presentation today on the topic animal cloning application and status in India. So now I request uh, Dr. Prem Singh Yadav sir for the presentation, please.
Thank you, Dr. Thank you very much. And thank you for giving this opportunity. Are you listening my voice?
Yes, sir. Yes.
Okay.
Yeah.
So I will talk about the animal cloning technology, status and scope in India. First thing, I always share with the participants wherever we get the opportunity that we should know exactly about what is animal cloning, what are the facts. So clones are not genetically engineered animals. Sometimes there remains confusion to the people that these are genetically modified animals, but these are not. Offspring of the clones are not clones. If we use the semen or the females for producing the young ones, then these are not clones. These are like the normal progeny. Aging is normal in clones. Some people have a perception because Dolly expired before time, so people have confusion that animal clones have shorter life, but this is not true. The same group which produced Dolly using the same animal cells produced more clones and they lived for complete life. The clones and their offspring products, milk and meat products, are safe for human consumption. This has already been declared in a risk assessment study by FDA in 2008. Even labeling is not required for cloned animal products. And many countries use animal cloning technology to improve the quality of animals for food production. In India also, two institutes, NDRI and CIRB, are working on these aspects from the last 14 years. And now we have about 18 clones, and most of the male clones are now adult and they are producing the semen, and even we have produced the progeny from the cloned bull semen that I will share with you in the coming slides. So these are the animal cloning company. It is the name of the company, country, and the animals being cloned. So you can have a look that dog, cat, horse, bovine, ovine, caprine, endangered species, camel, pig. These have been cloned, and buffalo has been cloned in India, China, and Thailand. 3 countries have cloned, but at present, India is the most leading country as far as animal cloning, buffalo cloning is concerned. Here, this is the slide which shows the procedural aspect. So how we are doing the cloning. So in this, there are two pathways. One pathway is for the donor cells and another is for the recipient oocyte. So we take the animals because now procedures are standardized to a greater extent. So we take the superior animal. It can be male or female, which we want to clone. We take very small tissue from this animal and grow the primary cells. The fibroblast cells are grown and preserved. These are frozen, and as per the needs, these can be thawed and can be used as donor cells. So one such cell is required for cloning to make, and this somatic cell is known as donor cell. On the other side, we take the oocyte. Oocytes are harvested from the ovaries, and ovaries we are collecting from the slaughterhouse. These are the visible surface follicles are aspirated, and these follicles are on the surface of the ovary. We collect the immature oocytes. These oocytes are having cumulus cells, so we need to mature these oocytes for 22 to 24 hours, and then matured oocytes we get. These cumulus cells are shedded out, and then there is zona pellucida. So in our cloning method, we dissolve the zona pellucida method with hyaluronidase enzyme, and then the part which is having nucleus that bulges out on one side. Once we remove the zona pellucida, the polar body or the part having the nucleus bulges out, and this is cut manually because we don't want to have the genetic information of the oocyte, because oocyte and sperm have n number of chromosomes. So this n number of chromosome of oocyte is cut and thrown away. Only the cytoplasm without the nucleus or without the chromosomes or without the genetic information is used. And two such cytoplasts are used and one donor cell, because the donor cell is a somatic cell which is having the 2n number of chromosomes. So this is sandwiched between the two cytoplasts, and with the help of an electrofusion machine, a minor current is given to fuse them and make them rounded. And then it starts growing, making 2 cell, 4 cell, 8 cell, and morula blastocyst. At blastocyst stage, these embryos are transferred into the synchronized recipients. Ipsilateral hole. In the ipsilateral means the side on which there is corpus luteum, we transfer the blastocyst stage embryo in the animals on that side. After 28 days, we check the pregnancy with the help of ultrasound, and those animals which becomes pregnant, they calve after a normal gestation period. So this is the method, and in this way we can produce or multiply or photocopy or generate the animals like the animal from where we started, the donor animal. If the donor animal is a male, then the clone will be a male. If donor animal is a female, then it will be a female. If it is a superior genetic merit animal, then your clone will be superior genetic animal. So this way we can multiply at faster speed. This is a vegetative propagation of the animals. So this way we can produce more animals in a shorter time, and best animals can be cloned. So So in 2018, when we started the program, there was 1% success in cloning efficiency. So our one of the aim was to produce the clone, and another aim was to improve the cloning efficiency. So we tried to improve the cloning efficiency also, in which one approach we used was the improved management of the recipients. We had a separate herd of 80 animals for as recipients. These animals were well fed, their all diseases were tested, and then ultrasonographically they were studied for their reproductive functions very regularly. And animals were synchronized as per the need. Then their heat was detected again with the help of ultrasound, and on the day of transfer, the corpus luteum was also checked. So this way, we tried to improve the management of the animals as well as some modifications in the protocols to make the clone embryos, and we could improve the cloning efficiency from 1% to 6 to 8%. So these are some of the major achievements which could be made during the last 4 years. That 7 clones of a single breeding bull were produced, and this is a world record of any species to produce such a big number of clones from a single animal. 3 clones of another single breeding bull were produced, 2 clones of another single breeding bull M2501 were produced. We, for the first time in the world, recloned a clone calf, which we produced in 2015, and 7 clones and recloning. Both these achievements are unique in its kind and have been recorded in the India Book of Records. We also produced female clone of the highest milk producing animal in our institute. So this way we have multiple clones. Another achievement, I told you that once we were transferring 100 cloned embryos to the recipients, there was expectation of 1 calf. So that was really very, very less, as well as if someone is transferring even 40, 50 and not getting a calf, so it is very frustrating for the scientist as well as not good for the technology. So we used 2 methods. One was, I have explained, that recipients were managed in a nice way, and another was to treat the embryos with genetic modifiers, and we could produce quality embryos, and this way we could improve the cloning efficiency from 1% to 6 to 8%. And still we are working to improve it, and our target is to take it up to 20%. We have also studied the age, growth, and fertility of male clones and found that these are similar to the non-cloned bulls. We have produced more than 100 progenies from the cloned bull semen, and at present we have about 750 artificial inseminations from the cloned bull semen. So we are now planning to have thousands of the progenies from the semen of the cloned bull, and whatsoever progenies we have already produced, they are showing normalcy and indicating that they are good. Their gestation, growth and health of the cloned bull progeny are normal. Some of the clone embryos have altered DNA methylation. That problem we have resolved. Sperm transcriptomics studies demonstrated that mRNA and miRNA are similar to non-cloned bulls. So this way we have produced, studied the fertility, compared with the non-cloned bulls, and also studied another parameters. Particularly, I will explain in the coming slides that the production of multiple clones was tweeted by Indian Council of Agricultural Research to PM, Prime Minister. And this has been also published as a news in scientific journal Current Science on 10th October 2020 issue. So I have already told that 7 clones and recloning, these have been getting quite good importance worldwide. Even Chinese people have invited me to deliver a lecture after this achievement. One achievement we made in 2017, that was to clone the SME buffalo male. So I brought small tissue from Guwahati and we cultured the cells in Hisar, produced the embryo in Hisar, transferred the embryo 100 kilometers away from Hisar in a dairy farm and produced a SME's buffalo clone for the first time. This is the first SME's buffalo clone and current science journal has given space on its cover page. And this has also been aired at the Rajya Sabha TV. You can see on Rajya Sabha TV is documentary of 4 minutes adding the keywords SME's buffalo clone. YouTube पर जाकर के आप you can see SME's buffalo clone CIRB. You will find a 4.5 minute documentary which is excellently presented because that was film was made by Rajya Sabha TV staff, a commercial people, so they have made it very good for education purpose. So these are the, I have told that this is a bull which is now top ranked progeny tested bull 4354. And these are the clones of this bull and this is reclone of you. So we can clone as well as reclone and all these clones are producing quality semen and we have 30,000 doses of cloned bull semen from this Hisar Gaurav which was produced in 2015 and we have now about 100 progenies from this and 750 AIs we have done which we are expecting to deliver in the coming months and we are having another project in which we are doing the artificial insemination with the cloned bull semen in new district of Haryana, which is an aspirational district, and our target is to do 4,000 AI in 2 years, so that we can get about 2,000 progeny and out of 2,000, 1,000 female progeny, so that its milk production with the animals from which they are being produced can be compared and a larger data can be generated so that this cloning technology can be utilized throughout the country and our government should can recommend this technology. These are the clone calf produced in 2021 and 22 at NDRI Karnal, the same project which we are working together. I have told that improvement of success rate of the live clone production. So there was some DNA methylation profile में कुछ था problem. So large number of pathways are affected by hypermethylation in clone related to IVF embryos. Among these major pathways related to embryonic development are integrin signaling pathway, WNT signaling, inflammation mediated, and chemokine and cytokine signaling pathway, hemoglobin releasing. So we have added some cytokines and we have improved these pathways so that we can get better embryo. Also, we had the Sanger sequencing of the selected genes of these regions. So for these hypermethylation, we have used, these are the reprogramming enhancing agents which help in so the treatment with the epigenetic modifiers, AZA, DC, TSA, and DAG improves the embryo production rate and the live birth rate that we have conducted the research and improved the cloning efficiency. We first, I told that produced Hisar Gaurav, the clone bull in 2015. And for 5.5 years, we continuously studied all parameters on these. What were the parameters we studied on this bull? Were the cell physiology, cell cycle, embryo development, epigenetics of cell and embryos, gene expression, pregnancy rate, health of the crown bull, fertility of the crown bull, and health of the cows of the crown bull. And in the conclusion, in a paper published in Nature Scientific Report, we have seen that the growth, health, and fertility of the crown bulls is similar to non-crown bulls. So this is a comprehensive 5.5 year study which has been published, and we are further working on this bull for more progeny. Fertility of the crown bull. This question is normally asked whether the semen quality of the crown bulls is same or not. So we conducted both in vitro, in vivo, and laboratory tests like post-motility and the other tests with CASA, that we could see that the fertility of the clone bulls is similar to non-clone bulls. The clone bulls which were produced at NDRI Karnal and CIR Visar, we used both the bulls from both the places. Also, we have studied the evaluation of the clone animals for their growth, health, and epigenetic status. Also, we have tested their volumes, spermatozoa concentration, total sperm count, mass motility, progressive motility, viability, acrosome integrity, and we could find that the 3 clone bulls, these parameters were similar to non-clone bulls, and this has been published in a good reputed journal, PLOS One. So these are the Chargoro AI-born progeny. This is Assamese buffalo bull, which has now become adult, and we have 4,000 semen doses from Assamese buffalo bull. This bull is named as Sachgoro, and these are the multiple clones of the M29. We produce dozens of progeny from the Sargoro, both at our institute as well as in a organized herd in a dairy farm, private dairy farm, with which we are having a memorandum of understanding to conduct the cloning research. Similarly, at NDRI Karnal also, they produced 2 clones in 2013 and 14. Now they have produced the progeny from the semen of these clones, and you can see the progeny. And they are also now planning to use the clone bull semen in Haridwar district, like our Innu district, to produce more progeny using the clone bull semen to generate more authenticated and reliable information. So we do all parameters, including the blood biochemistry of clone and non-clone age-matched animals. Also, we have done the evaluation of the telomere length of clone cow. When there is question of aging process, so age of the animal is decided by the telomere length. As the telomere length gets shortened, it may be in vivo or in vitro. Even if we culture the somatic cells and their telomere length gets shortened, the cells get senescence. They stop growing even after giving the best culture conditions. Similarly, if in vivo, in the body, the telomere length gets shortened, then the animal will not complete its normal life. So we compared the telomere length of clones and found that it was normal or even elongated compared to the age-matched controls. So this was an experiment which took about 1.5 years to standardize, and now we are regularly doing on all clones the telomere length study. So this, it gives an answer scientifically also that clone animals have similar age group, similar age as compared to another non-cloned animals. Spermatozoa global transcriptome analysis of the cloned bulls NGS data reveals that the expression levels of mRNA in spermatozoa of cloned bulls and their somatic cell donors are that regulate spermatogenesis, fertility, and early embryonic development are non-significant. Spermatozoa global mRNA of the cloned bulls that was again found similar and non-significant. So to give an overview of the cloned bulls available, you can have a look. All these are the clones which are available at CIRB, and all these which are visible to you are clones of a single bull M29, and you can have a look that they have been born out of different recipients. So weight of the recipient and behavior of the recipient may be different, but these clones are quite similar in their physical appearance, health, weight, and also now they have become ordered to produce the semen. These are other clone bulls which are available at CIRB ISAR. So I have shown you the 10 clone bulls which are available at CIRB ISAR. So 10 clone bulls means they can produce 10 lakh semen doses. So this way we can get enough semen if we clone the males to get many progeny, and if we clone the females, definitely we can have some progenies, but the number of progenies can be 10 to 12 limited. But semen can faster multiply the superior animals. That's why our emphasis is more on the clone males. And these are the clone bulls available at National Dairy Institute, Karnal, in the same project. So you can have a look that now we have about 20 clones and some pregnancy. So this way we can give good germplasm to the country, and at least some of the bulls which are in the progeny testing program or some of the bulls which are in the germplasm improvement programs can be produced through cloning technology and can be utilized at national level. So these are some of the salient achievements produced. This is a 4 years project because the earlier projects were going on at both the institutes. So produced 13 clones from the 6 superior breeding bulls and 1 elite buffalo, 7 copies of superior bull M29, 3 copies of the top ranked bull MU4354. Some pregnancies are going on and recloned a calf, improved cloning efficiency from 1% to 6 to 8%. Fertility of the clone bull was studied, and we have produced more than 100 progenies now till now, and 30,000, more than 30,000 semen doses. Established a somatic cell bank. Now we have about 60 top animals cell bank at our place, which can be used by other researchers. Any of you can have the cells from that for making any type of research. And male progenies of the clone bull Isargoro started donating quality semen. Female progenies of Isargoro have calved normal and they are in milk production, and even some animals are in the second calving. So this is an overall. So what we learned out of this work in the last 4 or 5 years, that multiple clone is successful in buffalo, recloning is also successful. Male clones have similar semen parameters and fertility comparable to non-clone bulls. Both male and female progeny of the clone males are normal. Gestation length, telomere length, all hematology, blood biochemistry, growth parameters, and proliferation of the cells of the clones are comparable to non-cloned bulls. So when you work, when there are some solutions, there are new questions also come. So now we are working on the whole genome sequencing to get more information. Mitochondrial fate in the clones, comparison of fertility of the more clones which are now in semen production. Why cloning is not successful? Equally successful in all animals, because in animals, in any species, as per the literature, there are about 25% animals. Even with the best efforts, it is difficult to clone those animals. So definitely there is some physiological barrier which needs to be studied, so that all animals can be cloned. And how to improve the success rate by another 10%. physiological normalcy of the clone embryos in spite of the fact that they look brilliant. Sometimes in the laboratory, we see that embryos are excellent, but in spite of that, they don't turn into pregnancy. So this also needs to be studied. And production potential of the clones, because it is a time-consuming species, so it will take another 5 to 10 years that the progenies which are coming out of the clones, whether they are producing the same amount or more milk. So what is the genetic gain in them? This also needs to be studied, and we are working in this direction. So, and also the implementation of the cloning technology for breed improvement. So we have written to the government of India so that this technology or the semen from the bulls or the clone animals can be used for production of the progeny and testing, and then recommending it for the national need. This project we have completed from 2018 to 2022 March. Now Government of India, looking in the progress which we have done, have given us another project for 3 years, and this is on evaluation of the semen characteristics and fertility parameters of the clone bulls and performance of clone progeny at larger scale. So these are some objectives. So now we are evaluating the semen and evaluation of the epigenetic and molecular changes. So this we are doing at large scale, and we will. This is started in May 2022. So this is a DBT funded project, and the title of the project is Enhancing Economy of the Livestock Farmers Through Using Clone Bull Semen, which I are talking that we are doing implementing it in no district. In this, we are doing artificial insemination using superior clone breeding bull semen, record AI consumption rate, calving and performance of the progeny, and also organizing training to make the people aware in the aspirational district. तो अभी तक जो इसमें काम हुआ है उसमें अभी तक मैं बता रहा था आपको 669 डेयरी फार्मर्स और बेनिफिटेड, एंड इसके अंदर हमने अब तक 750 482 से अब आगे बढ़ गए हैं क्योंकि अभी ब्रीडिंग सीजन चल रहा है। 750 एआई हम कर चुके हैं, और इससे जब आने वाले दिनों में क्योंकि सीजन है अभी अक्टूबर नवंबर दिसंबर में, वी आर एक्सपेक्टिंग टू हैव 2000 एआई दिस ईयर टू हैव मोर क्लोन प्रोजनी। तो ये आर्टिफिशियल इंसेमिनेशन में जून जुलाई के अंदर हमने की है, और वी आर चेकिंग देयर नॉन रिपीट अबॉर्शन और एनी अदर थिंग्स। So thank you very much for patient listening, and any questions which you want to know, I will be happy to answer.
Thank you very much, sir, for a very nice presentation, and in detail you have presented the information about the animal cloning, the success rate, and whatever the applications and achievements they carried out in India, that you have presented in a very simple and very nice manner with very good photographs. Excellent presentation, sir. Now I request the participants, you can ask the questions. Participants, they can write in chat box also, or directly they can ask the questions. This is very important topic as far as our certificate course is concerned, because our title is saying that enhancing livestock production and food safety through One Health approach. So for livestock production, this nowadays the cloning is very much important for this breeding purpose, for the improvement of breeding. So participants, please. So one question is there in the chat box. Dr. Vivek, you can read that.
I am not able to see the chat box. Can you
Yes.
read the question, please?
Through some light on cloning and food safety that is mentioned in that.
Yeah. Food safety, I have already explained that FDA has already conducted. You can have a report on the internet risk assessment studies on cloning. Risk assessment studies on cloning by FDA. So this is a document which has been published and put on the public domain by Food and Drug Administration USA, and they have conducted the studies at different locations, and this is a 1,000-page report, so which gives all truth and nails, all aspects about the safety of the cloned bull meat and milk on different locations, and they have declared it safe for human consumption. So that is also in 2019 and 20, European Commission has also declared it as a safe food. So they were reluctant earlier, but now they are also saying that this is a food which can be used. And you know that hundreds of the cloned animals, particularly males, are produced. There are private companies in many countries which are using, and there is no need of labeling. So the semen and meat products are being utilized by the population. So that gives us a clue that the animals are safe, and we in India are working on all production aspects. We can also find out the people who are working on the meat aspects are like our earlier speaker, and they can conduct the research on buffaloes also. So on other species, it has been declared as safe, but in Europe and America, because they are not working on the buffalo cloning, so the buffalo cloning, we are studying on these aspects. But physiological and biochemical parameters of the clones and their progeny, we have studied and found them normal. Okay.
Thank you, sir. Sir, one more question is there. What
Yeah.
are the legalities involved in cloning of animals in India? And
Legalities are, as far as legalities are concerned, now the projects which are approved to us are approved by the Government of India, both by National Agriculture Science Fund and Department of Biotechnology. And recently, our Director General ICR, earlier Director General, has written a letter to DADF that this technology should also be used like OPU IVF or IVF technology for improvement of the animals. So we have already written through our institute, then department, our ICR through DG to DADF, and their response is awaited.
Sir, one more question is there. Whether these legalities, they are same for the pet animals and domestic animals, or there is any difference?
Till now, we have written for the domestic animals, but once these will be, these should be for every species, and utilizing this technology for pet animals should be more easier as far as I think. And many people who are having the pets, they have contacted me to clone their pets because they have too much affection with their pets and they are not going to come in the food chain also. So there is, I think there is lesser risk, and there should not be much restriction, but still the clear guidelines are yet to be developed at the national level, in which the pets as well as the domestic animals both should be included.
Any other question from participants?
And we together and collectively have to do all this exercise because we are the people who are having the work and knowledge in this area. So even the guidelines and the other recommendations, we also organized a brainstorming session on this aspect at national level, in which all people of ICR and DADF and DBT were invited. And they have made some recommendations. On those recommendations, we are utilizing the semen in the field also.
Okay, so if no questions, then thanks a lot, sir, for your valuable time and sharing your experiences in animal cloning. Wonderful, the information you have provided, very recent, all these updated informations, and whatever the work done in India, in your institute as well as NDRI, that you have provided the information. Definitely, the participants they will get the benefit of this, because these participants from the across the country, from the one from the ICAR institutes as well as the SAUs, the faculty members and PG and PhD students, they will get the benefits, sir. Thanks a lot, sir.
Okay, thank you for this opportunity, and thanks to the participants for participating as well as listening. Thank you very much.
Thank you very much, sir.
So thank you, sir. So all the participants, you are requested to join tomorrow again at 11 o'clock. Tomorrow again we have 2 lectures. So we will meet tomorrow at 11 o'clock. All are requested to join at 11 o'clock tomorrow. Thank you.